Next, infectious disease, that is AIDS, AIDS, prevention of AIDS, acquired immunodeficiency syndrome. Acquired immunodeficiency syndrome. It is one kind of sexually transmitted disease. It is the sexually transmitted disease caused by human immunodeficiency virus, that is HIV. Here, acquired immunodeficiency syndrome. Here, the syndrome means group of symptoms. Group of symptoms called syndrome. Group of symptoms called syndrome. Here, this kind of disorder, a dangerous disorder caused by virus, that is HIV. HIV is called retrovirus. HIV is called as retrovirus, it containing RNA genome. It containing RNA genome. Therefore, it is the one that is called as retroviruses. This kind of viruses, this kind of disorder, basically, this kind of disorder first reported in USA in 1981. That HIV is the retrovirus. That retrovirus can be transferred from person to person. It shows transfusion. The transfusion of HIV shows this HIV can be transferred through sexual contact with infected person. Sexual contact with infected person. It can be transferred. HIV can be transferred through contaminated blood during transfusion process. Contaminated blood, and this virus can be transferred, can be transmitted through infected needles and syringes. Usually, this kind of virus can easily transfer from mother to fetus or child. Infected mother to child through the placenta. These are the mode of transmission of viruses. Viruses can be transfused through sexual contact with infected person. Can be it is transferred from contaminated blood during transfusion process through infected needles and syringes through the placenta from infected mother to a child. These are the mode of transmission of viruses. Next, high risk individuals. People who have at high risk getting infections. Anyway, individual who have multiple sexual partners. Shows what is that? High risk and drug addicts. The drug addicts who take drugs intravenously during such time easily HIV can be transmitted, particularly in the case of high risk individuals. And The individuals undergone repeated blood transfusion. During such time, easily HIV can be transferred. And children born from infected mother, these are the high risk individuals in the case of HIV transfusion or AIDS. Here are people who have at high risk getting infections through these reasons, or the individuals who have multiple sexual partners. During drug addicts and regular blood transfusions, the child born from infected mother. These are the reasons for transfer of HIV. The symptoms of HIV. The major symptoms of AIDS. The major symptoms include persistent fever, diarrhea, and weight loss. These are the major symptoms. And loss of appetite. Ulcer and skin rashes. And one more symptom, enlargement of lymph nodes. These are the major severe symptoms of AIDS. Acquired immunodeficiency syndrome. Persistent fever means continuous fever. Diarrhea means motion, and weight loss. Immediate loss of body weight. Loss of appetite, ulcer, and it is very dangerous symptom of the AIDS. Ulcer and skin rashes, and one more symptom, enlargement of lymph nodes. To the identification of HIV in the blood. One of the diagnostic tests is used. One of the blood tests is used to diagnose the HIV. One of the blood tests that is called as ELISA, enzyme linked immunosorbent assay. It is the abbreviation. Enzyme linked immunosorbent assay. It is the abbreviation of ELISA, enzyme linked immunosorbent assay. It is the method, it is the diagnostic test to identify the HIV in the blood or body fluid. One of the tests is used, that is called ELISA test. Next, come to life cycle of HIV. life cycle of HIV or retrovirus. Life cycle of HIV. Usually, HIV after the infection, HIV human immunodeficiency virus is entered into the blood or body fluids, and in a blood, one of the WBC, that WBC is called as monocyte. That monocyte is called macrophage. First, after the entry of HIV into the blood, the monocyte enters the HIV. Here, monocyte enters the HIV. Macrophages enters the HIV. Basically, monocytes or macrophages are infected. After the infection of macrophage, inside the macrophage, here RNA. It is always RNA virus. RNA is entered into the macrophage or monocyte. That RNA undergo reverse transcription to produce DNA. By the process of reverse transcription, here one of the enzyme is required. It is called as reverse transcriptase. It is responsible for the formation of DNA from RNA. Reverse transcriptase enzyme is involved. After the formation of viral DNA, that viral DNA is entered into a host DNA, and that host DNA produces, through the process of transcription, the host infected cell, the host DNA produces viral particles. It means RNA particles which are produced. That RNA particles covers protein. Then there is the formation of new viruses, new HIVs which are formed. Basically, the HIV multiplication takes place particularly in the case of macrophages. Therefore, macrophages which are considered as HIV factors. Monocytes or macrophages which are considered as HIV factors. Usually, HIV enters into the blood, and that HIV is entered into the macrophage, and after the entry, the RNA is modified into DNA. DNA is incorporated into the nucleus, host DNA, and again the RNA particles which are replicated, which are produced around the RNA particles. What is that? New viruses which are formed. Basically, one of the cells is involved for the formation of production of HIV. Therefore, here macrophages which are considered as HIV factors. After the formation of new viruses, these viruses which infect T helper T cells. That helper T cells which are damaged, which are destroyed due to the entry of HIV virus, and immediately person causes immune deficiency of immunity. The person causes number of symptoms. Therefore, the name that is called as syndrome. Acquired immunodeficiency syndrome is caused by one of the virus that is called as HIV, immunodeficiency virus. That is a retrovirus. Here it is the diagrammatic representation of life cycle of retrovirus, life cycle of HIV, life cycle of HIV. HIV is called retrovirus. Retrovirus. The retrovirus containing two parts, two regions. One is the genome, that is viral RNA core. Around the viral RNA core, presence of viral protein coat. Viral protein coat, that is called as a capsid. Here, it is the cell. It is the body cell, animal cell. The animal cell is covered by a membrane. It is called as a plasma membrane. Here, retrovirus HIV lodged on the surface of animal cell, on the cell membrane or plasma membrane, and only after the infection, after the lodging, only RNA particle is entered. Only RNA particle is entered into the cell. The remaining protein part, protein coat remains on the surface of a host cell. After the entry of this here, viral RNA is introduced into the cell, introduced into the host cell. Only viral RNA is introduced into the host cell. After the entry of this RNA, it is called as viral RNA. That viral RNA is produces viral DNA. Viral DNA is produced by the process of reverse transcription by using reverse transcriptase. Here inside the cell, inside the host cell, RNA is modified into DNA, viral DNA. Viral RNA is modified into viral DNA with the help of one of the enzyme that is called as reverse transcriptase. That after the formation of viral DNA, that viral DNA incorporates into the host genome. Incorporate into the host genome. Host genome means that DNA, viral DNA is entered into the nucleus of the host cell and integrated with the host DNA, and the host DNA undergo replication to produce new viral RNA. New viral RNA is produced by the infected cell. The new RNA, viral RNA is produced in the infected cell. It is the viral RNA. That viral RNA, with the help of viral RNA inside the host cell, with the help of a translation process, with the help of a ribosomes, around that viral RNA, the protein coat is formed. The protein coat is surrounds. Then there is a number of new viruses are produced. The new viruses are produced within the cell, within the infected cell. The new viruses which are formed inside the cell. The load of the viruses which are maximum inside the cell bursts open, then there is a release of viruses. Such viruses, new viruses, can infect other cells, other WBCs. Here, these cells which infect T helper cells. Totally, T helper cells which are destroyed by HIV. The immunity of an organism, the immunity of the individual is decreases. Person causes one of the very dangerous group of symptoms called AIDS. It is the life cycle of HIV, life cycle of retrovirus. Here, it is the retrovirus that should be containing a viral RNA core, RNA infection. With only that RNA is entered into the host cell. That RNA is modified into viral DNA. That with the help of reverse transcription, here the enzyme reverse transcriptase. That viral DNA is incorporated into the DNA of the host cell, and new viral RNAs which are produced around that new RNA, only the protein coat is covered, then there is a formation of new viruses. Such new viruses immediately infected with the TH means T helper cells. If the T helper cells which are damaged by viruses, immunity or body defense is decreases. This is the life cycle of HIV. Next, controlling measures of AIDS or preventive measures of AIDS. One of the organization called NACO, National AIDS Control Organization. National AIDS Control Organization introduced in 1991. NACO, National AIDS Organization, and other NGOs, non-government organizations, educate the people regarding what is that AIDS. Educate the people regarding spreading of AIDS. WHO, World Health Organization, started number of programs to prevent the spreading of HIV. WHO, World Health Organization, introduced number of programs to prevent the spreading of AIDS, HIV, making blood banks safe from HIV. Means HIV negative blood is used in blood banks. HIV negative blood is used in blood banks. And one more preventive ensuring only use of disposal needles and syringes in hospitals and clinics. Ensuring only use of disposal needles and syringes. And one more preventive measure, free distribution of condoms. Use the condoms during intercourses. Free distribution of condoms. And controlling drug abuse. It is one more measuring only that preventive measures of AIDS. And advocating safe sex and promoting regular checkups. These are the preventive measures of AIDS. NASO introduced 1991. Along with that, NGOs educate the people regarding spreading of AIDS. WHO started number of programs to control AIDS or avoid the spreading of HIV. Use HIV negative blood in blood banks, ensuring only use of disposal needles and syringes in hospitals, free distribution of condoms, and controlling drug abuse, advocating the safe sex and promoting regular checkups. These are the preventive measures of AIDS. So it is the diagram of retrovirus. It is HIV retrovirus, it containing viral core and viral protein. The virus infect normal cell. Virus infect the normal cell only after during infection. Only RNA is entered into the host cell. RNA is entered. only RNA is introduced into the host cell. That RNA produces DNA. Viral DNA is produced by reverse transcriptase enzyme. Viral DNA is produced by the process of reverse transcription. That viral DNA incorporate into the host genome, host DNA, host nucleus, and then new viral RNA is produced by the infected cell. It is viral RNA. The new viruses are produced from the host genome. Around that, protein coat is covered. The loading of viruses increases in the cell. The cell is burst open. Then there is a release of new viruses from the host cell. These viruses can infect helper T cells. It is the diagram of life cycle of HIV. Here there is a note. The infected cell can survive while viruses are being replicated and released out. It is very important. This diagram is very important for your future examination. Life cycle of retrovirus or life cycle of HIV.